కీ ఉంది
ఇందులో ఈ చేతిలో ఏం లేదు.
ఇదా బోర్డు ఇదా? ఏదో టాపిక్ స్టార్టర్ అండి. Good afternoon. I'll be giving you a demo on the Morgan's linkage concept. Morgan, as you are aware of, he happened to be that important scientist who had given us the idea of the crossing over part or even a mutation, or even get back to the concept of the most important thing is about the linkage concept. Now, according to the Mendel part, we are aware that Mendel had given information about the factors or genes which are responsible for the transmission or inheritance of a given character. But he could not conclusively explain some of the most important concepts of how the genes happen to be linked together. That is, as per the chromosomal theory of concept, he told that genes which are located on the same chromosome obviously will be inherited together. But as per the Mendel's concept, every gene is told to be that kind of factor which would be giving off its inheritance separately. I am talking about the independent assortment of the so-called characters. So therefore, as per the linkage concept of Mendel or the Morgan's concept, he had given us a pure light of ideas telling us that genes which are located on the same chromosome, obviously if they are very much close by to one another, definitely they would be linked together, which obviously means that the characters, if we talk about the Drosophila, the animal experimental model of the genetics, also called as the Drosophila. Now this Drosophila or the fruit fly, Morgan happened to study on the three important characters of the Drosophila, such as the color of the eye. Obviously, we are aware that the color of the eye would be in the two forms, wild form and the mutant form. And in case of the wild form, we are aware that the Drosophila is told to be having the character called as red eye, whereas the mutant one should be having obviously the white color, right? Now these alleles as such, or these genes as such, are present absolutely on the chromosome called as the X chromosome and very much close to one another. Very much close to one another. I mean to say, if I call this thing to be the loci of the character called as the body color, I would be calling this exact location to be the character for the color of the eye, which should be either this must be Y or Y plus, and this must be W or W plus. I mean to say, the plus mark indicates it's a wild character. So these two genes, as they are located very much close to one another, during the process of inheritance, these two things get inherited together. And we talk about another important feature is the shape of wings. Wings was denoted by a symbolical representation of something called as M or VG, vestigial or miniature. And usually the wild form is told to be having something called as M plus, or we can call it as VG plus. In other words, it means that this happened to be the allele which is responsible for inheritance of the character called as large wings. So large wings inheritance is located on this allele or this locus, and these two happen to be the alleles responsible for the color of the eye as well as the color of the body. So altogether, this comes under the chromosome called as X chromosome. During the linkage concept study of the Morgan, he happened to take two important crosses, say test cross number 1. In the test cross 1, he happened to study these two at a time, and test cross 2 happened to be that cross where he happened to study one of these along with the shape of the wings. conclusively speaking, in test cross 1, we happen to find the parental combination to be more for these two characters compared to the recombinants, right? Recombinants were few when compared to the parental characters. So parental characters obviously were more in number. I'm talking about the progeny of F2 generation. F2 generation had given more of the parental combination compared to the recombinants. In case of the test 2 or test number 2, he happened to find any one of these genes along with the gene which is responsible for the shape of the wing. He happened to find parental cross and recombinants almost in equal number. So this conclusively tells us that in case of the test cross 1, the parental number were more when compared to the recombinants, whereas the parent number and recombinants almost were equal. In case of test cross number 2 is just all because of. In case of the test cross 1, it implies that these two genes or these two alleles happen to be located too close to one another. So obviously they would be inherited together. When I talk about any one of these with the shape of the wing, because they are very much far away or more far they are, so it's something like inversely proportional to the linkage concept. More far they are, obviously they will be inherited separately. More close they are, they would be inherited together. We call this concept as the so-called linkage thing. So according to the Morgan's concept, and which is one of the important modifications to the Mendel's concept, telling us that the chromosomes which do contain the so-called genes upon them would be giving us the so-called characters. It all depends upon how close the alleles are. If the alleles are very much close by one another, definitely they would be inherited together. The more far they are, the more chances of they not getting inherited together. In other words, they happen to be the one which come under the non-linked kind of a thing. If they are too close by, they should be coming under the concept of the so-called linked characters. So linked characters and the one which happen to be the one which are very much far away should be the one which should come under the non-linked kind of a thing. So these are the experimental results which we came to know through the Morgan's experimental linkage concept. One more important thing. According to the Morgan, he told that this process of coming together of the alleles or coming together of alleles happen to be during meiosis. You are aware that during meiosis there is a process called as the chromosomes happen to duplicate themselves. And not only do they get duplicated themselves, they would be giving rise to the formation of something called as sister and non-sister chromatids. So if you talk about the sister chromatids and the non-sister chromatids, there is a concept of exchange over of the non-sister chromatids. We call this as one chromosome, this is another chromosome. Then I would be calling this as the sister chromatid of the first one, sister chromatid of the second one. It so happens that these two happen to be the non-sister chromatids. The non-sister chromatids as such have the property of crossing over themselves. That is, during the formation of the synapses, they would be coming together and they would be exchanging their chromatid segments. And if the chromatid segments do get exchanged, we happen to find this chromosome should be always the parental one, this should be the parental one, and these two come under the recombinants. So more chances of recombinants and the exchange of the chromatid segments along with the alleles is possible. It all depends upon how far the alleles are. If the alleles are too close by, this kind of the exchange of chromatid segments won't take place. Or if it takes place, it would be getting in a very less number compared to the test cross number 2. In case of test cross number 2, I'm talking about the allele, any one of these two along with this one, because they are very much far away located. So therefore, there is every possibility of this along with this exchange of segments obviously does take place. So more exchange takes place implies that more chances of recombinants. So therefore, recombinants are more possible in this case, but rather in this case, recombinants are very less. Now recombinants are very less. What does it indicate? It does simply indicate that the exchange of chromatids could not take place. So if the exchange of chromatids have taken place, it simply obviously tells us that the alleles or the genes are located very much far away on the given chromosome. More close they are, more chances of they getting linked together. Their segregation as such obviously would be very much different when compared to the segregation of the same alleles if they are located very much far away. Okay, so that was the linkage concept as per the Morgan's part. Thank you.